So welcome to the fourth Simulation Guided Trial Design webinar. What we're going to look at in this webinar is how we can use simulation and the results of simulation to help us select a futility interim and set the futility criteria in a phase 2 trial. So we'll look first at a nice simple phase 2 trial design, and with just two arms. And the question will be, well, how do we choose when to have the interim? And we've got a question on Q&A already. Oh, yeah. I need to promote Nick so he can help keep an eye on. I don't. There we go. Okay, sorry about that. In this webinar, we'll look at a simple phase 2 trial with just two arms for treatment and control. And the question will be, how do we choose when to have an interim for futility and how to set the threshold? And how can simulations help us do that? And then hopefully we'll have time to extend that example to a phase 3 where there's 3 treatment arms. Say we've got a medium, a low, a medium, and a high dose. And we want to extend our futility stopping into arm dropping, being able to drop any of those arms if they're looking particularly poor and unpromising. So we'll start with the simple case. And we're going to do this all through the medium of an example. So imagine you've got a phase 2 trial with a dichotomous endpoint. And let's say the response rate on control is expected to be about 20%, about 0.2. And the consensus is that it ought to be somewhere in the range 0.18 to 0.22. And the hope for target response rate on the treatment is 40%, about 0.4. But again, there's a little a range of expectation between, say, 0.36 and 0.44. As this is phase 2, we're not bound by regulatory control of time and error, and we choose to try to achieve a false positive target rate of 0.05 and a power of 90%. But if we look at these different rates possible, so on both control and treatment, they give us quite a wide range of possible sample sizes. We could have a control rate as low as 0.18 and a treatment rate as high as 0.44, which would require a sample size of about 52 per arm. If it's in the middle of our expectations, 0.2 versus 0.4, the sample size would be 88 per arm. And if it was the kind of worst of both worlds, the highest control rate, 0.22, and the lowest treatment rate, 0.36, we need as many as 178 per arm. So that's over a 3-fold difference just from this small range of uncertainty in sample size. Now, you might address that by including stopping for success and do a kind of a group sequential thing. We're not going to look at that here. We're going to assume that for success, we just want to go to the end, get as much information as possible on the treatment arm, given our sample size. But the company does want to be able to stop for futility if it can, if the treatment is clearly unpromising. Now we're going to make things a little harder. We'll say the time to endpoint is 6 months, so 26 weeks. That's obviously going to impair our ability to stop early, but the accrual is relatively low. It's about 3 per week, with about 12 weeks to get up, expected to get up to that rate. And for some of these sample sizes, as you'll see, that's going to require perhaps a year and a half, 2 years of accrual, which is 3 to 4 times the time to endpoint. So there is some limit. There may be some limited opportunity to stop early. And also, we've got to deal with an expected dropout of rate of 20%. So the first thing, So first thing we want to do is look at the sample size. And one of the ways we can do this, in fact, is to use variance and then look at the actual simulated power. So that's where we'll start with this example. The fixed variance. Here we go. This is the file. So we're going to start. So we have we don't turn anything fancy on. We have a 26-week endpoint. We just recruit continuously, and a positive and a response indicates an improvement. We ignore this maximum number of subjects because on the variance tab, I'm going to set up a number of variants, 6, with different possible sample sizes to simulate, from 200 up to 300 in steps of 20. And on the study arm tab, I've just got control and my one treatment arm that I leave with the default name of dose 1. From all the different possible scenarios I could simulate, from crossing rates of 0.18, 0.2, 0.22 on control, 0.36, 0.4, and 0.44 on treatment, I pick the lower two, 0.18 and 0.36. I pick the middle two, 0.2 and 0.4, and then a slightly pessimistic high control rate but middling treatment rate. So we'll say let's take a look at what the power will be over those scenarios, and I throw in a null as well, just to reassure myself that we are genuinely controlling the false positive rate to the extent required. On the execution, here's my mean accrual of 3 per week, and I've said there's a ramp up, and the ramp up completes at week 12. So you can see we've got some sort of there's the ramp up and then a level accrual. So it's going to be simulated stochastically. There will be some uncertainty around this, and it might take up to three years. But depends on the actual sample size in practice. But the quantities of interest, I'm really only interested in one. That's the posterior probability of being better than control. I'm going to specify the posterior probability being better than control at the treatment with the greatest response. I've only got one treatment, but this means that I won't have to change it when I add in additional arms. So I also keep the probability of being having the maximum response. So this is my decision quantity, posterior probability of being better than control at the arm with the highest probability of having the greatest response. And then the design is very simple. I've got a roughly uninformative prior with a mean 0, a standard deviation of 2, which is pretty uninformative in the log odds space. And doing simple one-to-one. Oh, and that prior is applied to both arms. I've got a one-to-one allocation ratio, and I've got a simple success criteria that the posterior probability being better than control has to be better than 0.95. And that should give me my error, my false positive rate controlled at 0.05. And now what you can see here, in fact, is each of these different response scenarios, null, 0.18 versus 0.36, 0.2 versus 0.4, 0.22 versus 0.4. Each one of those has been crossed with each of the different variants, variance 1 to variance 6. Which of these different sample sizes? Variant 1 is 200, and variant 6 is 300, with them going up in steps of 20. And then we can simply look at the proportion of overall success, and we can see under the various nulls, things are by and large kept under 0.05. There's a couple of blips above, but that's just, we've only got 1,000 simulations, so that's just some random sampling, taking it over. And then we can see that by and large, as we would expect, the power goes up as the sample size goes up for the different variances. And you can see the sample size reflected here in the mean number of subjects. Because as up till now, we're not doing any early stopping. These are all fixed trials, though the average sample size is the maximum sample size for each scenario. And if we can then simply tabulate those, and we can see, 300, where we've got more power than we really need. 260, we achieve, not quite, 0.28. Yes, at 260, we've almost got 90% power in all scenarios. And in 220, we've got 90% power in our sort of median scenario, 0.2 versus 0.4, and then a little bit less power, but over 80% in these other two scenarios where the responses are somewhat different. And let's say the team are keen to keep costs down and speed up, so we go with 220 on balance being just about enough. And of course, this is somewhat larger than the simple sample size calculation for 0.2 versus 0.4 because we've got this 20% dropout rate. So this, on average, is in practice a sample size of around about 180, which is pretty much exactly what the sample size calculation was. So we take our selected sample size and we just confirm that. So I've got my fixed all VSR. So now taken the same file and I modified it by putting in my selected sample size here. I've turned off the variance and I've added in the whole range of different virtual subject response rate combinations, null at 1.8, nulls all at 0.2, nulls all at 0.22, control of 0.18, treatment of 0.36, and so on up to a control of 0.22 and a response of 0.44. Everything else has remained the same, including the success criteria, allocation, and the prior. And we run, for the sake of it, just at first instance, 1,000 simulations of each, and we confirm that by and large, we're getting over 80% power, except in the very worst case of the highest rate on control and the lowest expected rate on treatment, then our power there is only about 66%. So assuming we're happy with that, the question is how are we going to set our futility? Let's see, there's just a question on the dropouts, on the execution, on the dropout rate. The simulation of dropouts is completely at random. You can have no dropouts, a dropout rate per dose, or a dropout. If we had intermediate visits, you could set out the conditional probability of being a dropout at each particular visit on each particular arm. But we don't have any dropout dependent on observed or unobserved response. So now we want to look at what we might do to add in an interim and some futility. But to do that, what we do is we take this file, we save it, so we keep everything the same, but we now enable adaptive features so that on the design tab, we can now specify some interims. And what I'm going to do is create interims every 20 subjects complete. So from 20 up to 200, and of course we finish at 220. So we've got these 9 interims. I could then specify a futility criteria or a success criteria at any one of those interims, but I'm not going to. I'm going to let it simulate all the way to the end. And then what I'm going to do is I'm going to output what we call the weeks files, where a weeks file is a file for each simulation that shows what happened at every interim. So as you can see, I've got results rather than do every single scenario, I've picked what I think are sort of 4 representative ones. One of the nulls, one where the response is low, 0.18, and the treatment response is 0.36, the median one, 0.2, 0.4, and the higher control rate, but still 0.4 on the treatment. And I've simulated them 1,000 times. If we just look in that folder, you can see we have a weeks file for every single simulation, and I can pick any one of those. I can open it up. Why there we go. Right. And now for every single interim, Fax has written out for us and recorded what the state of the trial was at that point, how many subjects overall have been recruited, how many have been allocated, the estimate of mean response, the raw response, the number of subjects complete. These are the number of subjects with complete information. Oh yes, I'll just give a little note on that. What I selected was to time the interims in terms of the number of subjects who had the opportunity to complete. Now, the number of actual subjects that will have completed will be less, because some of them, 20%, roughly 20% of them, will have dropped out. So my actual number of completers is 31 and 34, but the subjects who'd had the opportunity to complete at this point was 40 on each arm. So this is my interim at 80 subjects for the opportunity to complete. The thinking behind using that is that operationally, it's much easier for the team running the trial to know when this interim is going to happen. So as the endpoint is 26 weeks, as soon as we've recruited subject 80, we know when that subject and all the subjects before that, so in other words, 80 subjects will have had an opportunity to complete. It'll be 26 weeks after subject 80 enrolled. And so that's when the 80 subjects with opportunity to complete interim will take place. So we've got plenty of time. We know when that date will be, and it gives us plenty of opportunity to plan that interim, to make sure that the DMC folks will be able to convene and review the report, that the statisticians in the independent statistical team will be able to meet and process the data, and that the data management team will be ready to pull the data out of the database in order for that interim analysis to occur. Whereas if you're doing it on 80 subjects actually complete, then of course you're constantly waiting. Has the subject dropped out? Will we be able to observe them, and so on? And you're not sure right up until almost the day, perhaps, that you'll actually be able to perform the interim. Obviously, this introduces, in sort of group sequential terms, an awkwardness that you don't know precisely how much interim what percentage of information you would have achieved. But we're not doing group sequential here. We're doing a slightly more approximate scheme that we're going to tune just through simulation. In this particular, I just noticed the question there. In this particular example, I'm not simulating patients having intermediate visits and us having intermediate information on a patient, which would allow us to impute their final endpoint. So in this instance, all we would have is their baseline, and a dropped-out patient is a lost patient for whom we're going to ignore that data. But so we have all these weeks files. I deliberately made sure I was generating a weeks file for every single simulation, and we can use those to post-process to look at how we should extend our design to add in interims for futility. So that's what we have done. And the first graph we're going to look at is just to give us a sense of how the quantity of interest we're going to look at, the posterior probability of being better than control, how that performs over time during the trial. So if we look at the null first, we explore the results, we look at the graphs for this scenario, and we look at POIs at interims. And what this neat little graph does, it's relatively new. It's first appeared in FAQ 7.2, I think. We've updated a little bit since then, but this basically functions the same way. What this does is it plots the first 100. If it did every week's file, the chart would just be a sort of dark mess. But here we can still just about make out what's going on. So each line here is a single simulation. And for each line, what it's doing is it's plotting the value of that quantity of interest, in this case, the posterior probability of being better than control, at each of the interims until that particular simulation finishes. Here, none of them stop early because I haven't set any early stopping, and they all reach the end. And you can see, because I've selected the null, that the vast majority end up as failures, and just a few type 1 errors up here at the top are successes. And you can see that under the null, this posterior probability kind of ranges over almost the whole range of values from 0 to 1 at almost every interim. If we look at one of the alternate scenarios by way of contrast, here we can see they're much more concentrated at the higher values. Not so much at the very early interims where data is sparse, but as the amount of data grows, so you can see more and more of these lines. Well, even the outliers have got a fairly high value, and the bulk of them are up here at the top towards 0.95. So if you consider those two graphs kind of overlaid on each other, you can see that there's a sort of a space down here where if we put in some sort of boundaries somewhere on some of these interims, we would clearly catch the simulations under the null and could stop them without hardly ever stopping a simulation in this particular alternate scenario. And even if we do catch one or two of them, then they're likely to have ended up null anyway. But we want to do something else as well and just look at what data we have in each scenario. We don't have an average one of these, but I can do it per simulation. And this plots 4 things here. It plots the number of subjects who've dropped out, the number of subjects who've completed, and the number of subjects who've been enrolled. And something this brings out, which we might not have appreciated if we hadn't looked at this, is that actually from those 9 interims I've added, from interim 6 onwards, actually, we're fully enrolled. And if we just look at a few other simulations over there, we weren't fully enrolled to simulate to interim 7. But between 7 and 6, and there's one even earlier, we're fully enrolled. So this area here, which was looking sort of particularly promising for an interim futility, well, we could stop perhaps pretty well for futility here, but we're not going to save any sample size because that interim is going to occur as we're just in the follow-up phase. So it might save some time. It still might be worth doing, and we'd save some costs, but we're not going to save sample size. Now this kind of gives us, I think, reassurance that actually some stopping for futility without much reduction in power is possible, but perhaps not so sure how beneficial it'll be, and we're not quite sure, particularly because this is only 100 simulations, exactly where that threshold should be. So we're going to turn to yet another graph. I'll show you the actual graph. This is, it doesn't transfer well to the slide. This is a thing called the interim versus final scatter plot. So let's look at the actual one. It actually looks a lot better for real than it does there. So let's close a couple of those graphs. So here I am. I'm still looking at my fact simulation with 10 interims and no early stopping. I now use the across scenario graphs. What I want is this interim versus final scatter plot graph. This is a quite complex and extremely powerful graph. It's really rather wonderful. Right, so what we've got going on here is in the graph, we'll come to this control panel in a moment, but on the graph, we've got a scatter plot and some stacked bar graphs. The stacked bar graphs is one per scenario, and the different sections of each bar and their different colors represent the proportion of simulations in that scenario and their outcomes. So here in the light green is early stopping for success. Dark green is final success. These light blue would trials that would be stopped for futility, but if they'd been allowed to continue, would have been successes. The purple band are trials that were stopped for success, but would have gone on to be futile. The dark reds are trials that went to the end and were futile, and in this case, small patches of light pink are those that stopped at the interim for futility. And all the individual simulations from those 4 different scenarios are plotted here on the scatter plot with a point per simulation. And along the x-axis, we see the value of the quantity we're evaluating to decide whether to stop or not. And then the y-axis, we're seeing the value used to determine final success or not. In this case, both cases, it's the posterior probability of being better than control. And on this control panel, we choose the quantity that we look at for the x-axis, the quantity we look at for the y-axis. In this case, we're using the actual whatever axis decided in the simulations. But we could actually override that and set different success and futility criteria. But we're allowed to set manually the success and futility criteria at the interim. That's why this plot is currently showing some early successes, even though I haven't included that in the design, because it's allowing any simulation which saw a value of greater than 0.9, that's just happens to be its default, to be stopped early for success. So we'll stop that by setting it to 1. We'll leave that at 0.2 for the time being. We'll leave it at interim 2, and we'll redraw. So the light green has gone away because I've made it impossible to stop early. And now we can concentrate on whether we like this futility boundary. So we can see the futility boundary is at 0.2. So everything on this vertical line and to the left will have been, every simulation there will have been stopped for utility, and all the light pink ones represent ones that would have been futile anyway. So those are good. That's us correctly stopping a trial for futility. But up here, we have a whole bunch that are light blue because they would have gone on to be successes. So they represent some degree of reduction in power. We've made a type 2 error at the interim. We can perhaps see get a better sense of the proportion here. As you can see, we in each of these scenarios, we at this threshold for early futility, we'd be losing sort of 3-4% power in each of those scenarios. So if we were going to stop as early as interim 1, we'd probably want to lower that threshold. But also interim 1, we've got very little data. We saw on the quantity of interest plots over time that they were, even under the alternate, they were quite spread out. So maybe interim 1 is not the interim we want to be first stopping at. So if we move it on to interim 2, you can see this population of type 2 error has reduced. Those points by interim 2, their interim values have improved or increased, and so those points that were type 2 errors now shifted to the right, and they're now the other side of this 0.2 boundary. And you can see the amount of type 2 error has correspondingly decreased in each of our alternate scenarios. And we can reduce it further by being a little more cautious. Perhaps at that interim, we want to change the threshold to just 0.1. And now you can see we've just got a very tiny number. And we can repeat that. We can look at further interims, let's say interim 4. And now at interim 4, again, you can see the green dots, those that will be successful at the end. We've not stopped early for futility. And now much more crowded over towards the right. And we've just got a small scattering over here. And we could probably almost certainly be prepared to have a more aggressive futility boundary, maybe at something like 0.3. Which introduces this tiny amount of type 2 error, extra type 2 error. But now we're getting not a massive amount of early futility stopping, maybe 30%, but at least enough to start making a difference. And we can also see that we could probably get quite a bit more early futility if that was really important to us, with only a little bit more type 2 error. So we maybe push the boundary up to 0.4 or even 0.5, depending on the needs and the pressures on this particular trial. So what do we decide? So let's say we decide that we have interim. What we're going to use are, out of those 10 interims, what we called interim 3, interim 5, and interim 7. And I've using that process that you've just seen, I've decided that we've got acceptable levels of additional type 2 error if I set those thresholds at 0.3 for the first one, 0.5 for the second one, and 0.7 for interim 7. And those will respectively, those will be at when 80 subjects have had the opportunity to complete, 120 and 160. But also note at this point, we'll be actually fully enrolled. So any stopping at this interim is saving us time and operational resources, but not sample size. So take this facts file, three interims, here we go, and I modify it. So we've still enabled our adaptive features. We've still got our cap at 220 on ARM. We've still got all our VSRs, same accrual and same dropout, same quantities of interest, same non-informative prior, same one-to-one allocation. But now we've limited our interims to those three that we selected, 80, 120, 160. And we now have these three tabs on the success futility criteria. In fact, you don't actually get all those by default. You have to say, I want the tab for interim 3 and hit create. And then at each one, I've enabled early futility and I've added the simple criteria. But the posterior probability being greater than control at the greatest response. If that's less than 0.3, then we're going to stop early for futility. At the second interim, that's the same, but it's less than 0.5. Another third is the same, but less than 0.7. Unfortunately, doing that. Oh, my simulation results, because it thought I was changing something. So let's just bring them back. And we can see the nulls are controlled at around about the right rate. Power still looks pretty reasonable. The amount that's stopping early for futility is obviously quite small because the sample size is nearly 20. And there we go, there's the full set of results. So there's our early futility stopping, over 70% in the null cases, and otherwise it's less than 5% typically. And if we stack those up against the power for the fixed all VSRs, look at the proportion of overall success, we may be down by percentage point to, in some cases, no reduction at all. Of course, what we're hoping to do is really only make a difference here. So if we pick one of these nulls, you can see the time course for futility stopping, and we can see that from about just over week 50 onwards, the futility rules cut in, and we get to stop in about 70% of the time, as we saw, with a good 30% stopping around about week 50 to 60. The sample size, you can see we've got the two stops at the first two interims, sort of 30% at the first interim, about 20% at the second interim, and then, of course, no change in sample size at the third interim because we're fully enrolled. And so we've got a reduction in the expected sample size of about 10%, and reduction in mean duration should be quite a bit bigger because we not only stopped enrolling, but we won't bother. We stopped for futility, so the duration doesn't include any follow-up. The assumption is we wouldn't attempt to fully follow up the subjects as on the grounds that wouldn't be terribly interesting. So we've got a saving in time of about 25 weeks. But of course, that's not speeding up our development program. It's speeding up how quickly we stop our development program and switch our resources to something else. So now what happens if we have 3 treatment arms? So we have to extend the scenarios we're going to simulate over. I'm going to assume roughly similar degree of uncertainty in what the rates are going to be. But now I have to also make some assumptions over what the relative response will be of the different treatment arms. I've decided that for each of the different, well, there's a typo there, that should be 1.8. For each of the different control rates I'm simulating, 0.18, 0.2, 0.22, I will have 3 different shapes of response across the treatment arms. The first one, a kind of a linear increase, so the high dose has got the greatest treatment effect. Then one where the medium dose has the highest treatment effect, but the treatment effect falls off at the high dose. So, perhaps the way we're dealing with intercurrent events like termination, use of rescue medication, failure to tolerate the drug, and those sort of things, we're assuming those are going to count as failures. And so we can't assume that the higher dose is going to be better than the medium dose. It may have more problems and more intercurrent events that actually bring down the score. And in extremis, maybe even the medium dose is lower score than the low dose. And so the third of the scenarios has the highest response at the low dose, and then sort of a linear decrease in response down through the medium dose and the high dose. And then that pattern is repeated at each one of these different basic control response rates. Now we've got 3 arms, so we adjust the success criteria to from 0.95 to 0.983. Here we're making a sort of the equivalent of a Bonferroni correction. So the Parke threshold is 1 minus 0.05 divided by 3. But I can close now these with just 2 arms. And now we've got the fixed 3 dose of the SRs. So we've done a similar sort of use of the variance to find an acceptable overall sample size, which we've chosen to be 440. And this again, we still got the null controlled at 0.05. And we've got perhaps a little bit lower power now from 0.75 up to 0.9. And the other thing we need to worry about, so in the VSRs, in each one of these, I've checked which of these in the scenario, which one is the arm I would want picked, which is, of course, simply the one with the highest response. So in the simulation results, as well as the power, we want to also look at the proportion of times we're successful and we picked the correct arm. And inevitably, that, of course, is somewhat lower than the overall power because we've got something like a 15 to 20% chance, depending on the scenario, of being successful but picking the wrong arm. And once again, we can look at the quantities of interest over time. But for picking the arm and arm to drop, the advice from Kurtvili is that the best quantity to look at is its probability of being the max. So what we do now is we repeat our trick of putting in interims but not stopping at them. So 10 interims with 3 doses. So here we've added all these interims. Once again, we've got no stopping criteria. We've also added in some new quantities of interest for making decisions, which is the probability of being the max for the low dose, the medium dose, and the high dose. Not because I'm exactly going to make a decision based on these specific quantities, but so they're available in this plot. And now we can look under the null. No, actually what we really want is just to look at one of these alternate scenarios. And I'm going to compare, I want to look at the quantity of interest at interims. So the probability of being max at the low dose. So this is in this interim, so in this scenario, this is a good dose. So this is one we don't want to drop. So we're looking at, well, down here, what sort of level of threshold could be set where we're not going to drop this arm too often, compared with, say, the high dose, which has got the, which is what we do want to drop. And there you can see indeed that if we had a nice low threshold, we would actually catch the and drop this dose, the maximum dose, which in this scenario has the lowest response, and be able to drop it. So that looks quite promising. And just to reassure ourselves, a low dose, it's better if we just zoom in on the range of interest. So maybe we'll cap the y-axis at 0.2. And you can see indeed that we've now quite a few of the plots have disappeared. And if we had a threshold of, say, 0.1, we'd stop a few, but it wouldn't be that many. And it's quite likely that those would have ended up not being the maximum dose anyway. Not the dose with the maximum response, even though that's what they were being simulated as. The date we would have been unlucky and had data that showed them odds being lower. Once again, we use the interim information over time, and here we now see that because of the much larger sample size, because we've got the extra arms, actually, if we have these 9 intermediate interims, the full enrollment isn't until just before interim 9. So we can have interims later, let's say interim 7 and 8, which we could use to drop an arm, and we would still be actually potentially saving some sample size. There's one limitation in facts, which is that for simulating an arm dropping rule, it's limited to a single rule. This is a little bit unfortunate, and we need to address at some point. But we can only set one threshold, and that same threshold used at every interim. So let's say we keep interims 2, 4, 6, and 8. We drop at most 2 arms. There are different things we could do when we drop an arm. We could keep the sample size the same and reassign the subjects across all arms and across control. We could keep the sample size the same, but now with the freed up subjects could just be assigned between the treatments. Or the third option, we could reduce the study size. We says, well, we've dropped an arm. We no longer need to recruit those subjects that would have been accrued to that arm because it's not there anymore. So we drop the overall sample size. Obviously, if you reallocate the subjects to the other arms, then you get a little bit of an increase in power, but you don't save any time or resources. Whereas if you reduce the study size, we're not going to improve the power any, but we might see a reduction in time and a reduction in sample size. And in practice, the reduction in sample size is kind of the more sensitive metric than the increase in power. So although the option is still there to do the other thing, we're going to reduce the study size because it's the most sensitive way of showing the effect of the arm dropping. So we now go down to 4 interims. So we've still got our sample size of 440. Everything else is staying the same. We've got our adaptive features enabled. We've got our 3 arms now, low, medium, and high. We've got our various different responses that I showed you the table of. The execution is still the same. It's still speed per week with a bit of a ramp up. Obviously, with the larger sample size, that's taking longer to enroll, now nearly 3 years. Here's something I didn't fix. Oh, that's interesting. Oh, okay. Because I was basing this on the previous fax file with just the control in the one arm. I've extended that to add in the other arms. What I had missed was this has extended the table for the dropouts, and I have not entered 0.2 and 0.2. So these numbers are now going to be a little bit different from what I was expecting. So let's see what happens. So here I've set up the 4 interims. Oh, sorry, I'm just showing the allocation here. So here's the arm dropping rule. Here's a more conservative option to only drop if an arm has the probability of being the maximum of less than 5%. We've decided we always want to go to the end if we can. So we're only going to drop up to 2 arms using this rule. If necessary, in other words, if 2 arms have exactly the same probability, we'll drop the lower dose with the arm with a lower dose. And having dropped the arm, we're going to decrease the study size. For our interims, we've picked the one at 80, 160, 240, 320. And we have also repeated our process of looking at a futility rule. And so we've included an early stopping futility rule. Using the graph we saw the symbol case, and we're stopping at the threshold of 0.3, 0.5, 0.7, and 0.8. And we'll now have to resimulate those. I won't do 10,000. Been doing that just to check my time on error. We'll just do 1,000. So you can see our sample size is somewhat reduced from 440 by the arm dropping. We can compare these numbers to the fixed design. We've got a little bit of loss of power from 0.9 to 0.86, 0.78 to 0.7576, 0.88 to 0.82. And we go right to the far end to check portion of correct arm. This is important in case we've been dropping the wrong arm. And we have got some reduction here. We've got a reduction of about 4%, 6%. Again, 6%. So we have got some reduction in this selection of the correct arm. That's interesting. That reduction, that the correcting the dropouts has changed that story somewhat. That loss wasn't so bad before, and not surprisingly, because I had got more data. So we might judge this as being unacceptable. And I guess we've got two. If we say, well, we've lost too much power and lost too much probability of selecting the correct arm, we either want to make the arm dropping less aggressive, the futility less aggressive, or give ourselves a larger possible sample size to aim for. So the expected sample size should still be less than 440, but allow it to go higher if necessary. Also, this one will have the same problem on the dropout. So I'm not doing a like-for-like comparison, am I? These will be very quick because they're just fixed trials. So actually, maybe the fall in power isn't so bad because once I fix the dropout stimulation, it'll be equal. And the drop in power is now more like 2%, but possibly my selection of total sample size is off a bit because I haven't got the correct simulation of the dropouts. So one of the hazards of simulation, well, too easy to make a little error like that. It's always worth tabbing through and making sure everything is set as you meant. Of course, possibility of making such an error is even greater when you're writing R code, and it's a little bit less if you're running facts. And of course, you've got the advantage if you're using facts that the simulations are pretty fast. So if it's much easier to recover from the error. Okay. I think that's all I wanted to show. One more thing I would do in this example, if I had a little bit more time, is look at the overall success. We know a Bonferroni correction is over-conservative. It seems to be spot on 0.05 here, but we've only got 1,000 simulations. If we made that 10,000, we might be able to find that that change in the success futility was slightly greater than we needed, and we could back off a little bit and use a success threshold of more like 0.98 instead of 0.9833, or maybe even 0.97 something, and that would recover us some power here. So yes, now we've got 10,000 simulations. We can see we now get a false positive rate of only just over 0.04. And if we've got a nice little graph we can use called Explore Final Success Criteria. And if we use the target dose is the PR max. We've only got one quantity of interest to evaluate. Here's the posterior probability of being better than control. We can set a target rate 0.05, and it tells us, yes, that if the threshold was set to 0.9792, so probably set to 0.98, just to be a little bit on the safe side, we would achieve a false positive rate of 0.05. And if we wanted to be even more definite, we could run 100,000 simulations here and repeat the exercise. And that might get us just another way of getting a little bit more power back. But I'm at the end of time, so thank you very much. And I hope that's been interesting. Next week, I will be away, and it'll be Kurt Veely presenting, and it will be still on phase 2, but now he'll be looking at dose response modeling. And I think, given some of the work he's been doing recently, I hope he gets a chance to include that, and it should be really interesting, I think. So, yeah, enjoy, and I hope you can make next week as well. So, thank you for your attention. Thank you for attending. Goodbye, everybody.